Hello, class. We are going over special senses today. I won't even turn on the camera. We're going to just jump right in. We have already looked at the anatomy for nervous system and special senses. We've done the notes for nervous. And so, special senses is a continuation of the nervous system, in that we then have specialized kinds of neurons that we refer to as receptors. So again, we start in the eye. You know, the eye is for vision. The occipital lobe is dedicated for vision, right? We talked about that already. So we have structures around the eye that protect the eye, that lubricate the eye, that move the eye. And so we have the eyelids on the front of the eye or anteriorly that have eyelashes, and those things are to protect the eye. So the eyelids protect the eye and close when something gets near the eye. It's a highly reflective area where you have high reflexes for blinking to prevent stuff from getting in the eye, to protect the eye. And the eyelashes also protect against foreign material getting in the eye. Associated with the eyelids, you have structures that also lubricate the eye. Now, I think on the anatomy, I kind of backtracked on something and showed you the lacrimal gland, which is superior and lateral. This sends tears across the eyes, and those tears go across the eyes constantly to wash things away and to keep the eye lubricated. The tears also have somewhat of an antimicrobial effect or antibacterial effect. The eyelashes have meibomian or tarsal glands or ciliary glands associated with the eye, the eyelid, actually, the eyelashes, and they secrete substances that help to lubricate the eye. So these are modified oil glands and modified sweat glands. Then you have the conjunctiva, and the conjunctiva is a thin tissue that's over the surface of the eye and also lining the inner aspects of the eyelids. It also secretes mucus that lubricates the eye. This particular slide doesn't say it, but you also have something called a caruncle. I'm going to zoom in on this a bit. And again, if you need this bigger, click on those 4 arrows out so you can see all of those notes more clearly. And also, you have right in this corner, I'm going to put an arrow, something called the caruncle. And it's not on this slide, and I don't know if it's on the next slide, but I want you to make note of it. Car and then uncle. And that also secretes stuff to lubricate the eye. And we'll look at the picture in a second. I just want to give you time enough to see the written portion. And so then here you see the caruncle. They say lacrimal caruncle. You see that term here. Okay. And then we'll look at the various layers of the eye here. This is a side view, and we'll get into the muscles. We'll get into the layers. We've already looked at those things, but we'll talk about their responsibilities shortly. So here, let's zoom in. This shows the lacrimal gland, which is superior lateral. And then there are ducts that come this way, and tears rush across the eyes like this. And then there are openings in the inner part of the eyelid, the most medial part, here and here. And you see, let's zoom in a little bit more. They lead into these canals here and here. Okay. and then this sac leads into the nasal passageway. So if you're crying and your nose is also running, right? You're bawling your eyes out and your nose is running. Your nose is not running because it's not. Your nose is running because you've overstimulated these lacrimal glands. Those tears are washing across your eyes and going into these puncta. These are called puncta. One is a punctum. And then it drains down into the nasal cavity. So what happens is these little openings here, here, and here. Oh, I got the wrong utensil. Tool. Here and here are like little drains. And so what happens is when you flood the drains, tears run down your face, right? So the tears are constantly washing across your eye, and the intent is to go into these little drains. On a regular basis, that's perfectly fine. That's what happens all day long. But when you overstimulate that lacrimal gland and you cry, then you flood the drains. And so then they can't handle all the tears and tears run down your face. But the tears are actually produced up here. Okay. Also on this picture, we see meibomian glands, and they're associated with the uh, eyelashes. They produce kind of an oily film that goes across the eye. And sometimes, if you have dry eye, it's because these meibomian glands are not active enough. Because that oily layer that they secrete helps to prevent your eyes from drying out. As you see here, it says it slows down evaporation. And so, if you're not producing enough of that secretion, excuse me, um, your eyes can get dry. Backtrack a little bit here. Okay, so this is looking at the side view of an eyelid, right? So here's your eyelash here, right? And then here's a meibomian gland inside the eyelid, just behind the eyelash. And you notice there are other glands. We won't even talk about all of these other glands, but you have gland of Moll, gland of Zis. All of these help to lubricate the eye. You have um, the conjunctiva here, which is the lining on the inside of the eyelid, and it also lines the surface of the eye. Okay. So let me go back for 1 second. I may not have said that this gland of Moll is the ciliary gland. Gland of Moll is the ciliary gland. Okay, we didn't name the gland as this, and you don't have to worry about that. But the gland of Moll is the ciliary gland, and so these glands produce a lubrication for the eye as well. Okay, now let's go to the muscles of the eye. We talked about these before. The lateral rectus is cut right here. Inferior rectus, superior rectus, medial rectus. Maybe I'll change the color here. The lateral rectus is cut. Inferior rectus, medial rectus, superior rectus. They move the eye in the direction of the name. So the lateral rectus moves the eye laterally. Medial rectus moves the eye medially. Inferior rectus moves the eye inferiorly. Superior rectus moves the eye superiorly. Okay. And then we have the inferior oblique moves the eye up and lateral. And then the superior oblique muscle moves the eye down and lateral. Okay. Notice our superior oblique muscle has a tendon that runs obliquely, but the muscle itself runs almost um, parallel to the rectus muscles. And then here's my optic nerve. And let's zoom in on this picture. Well, we can zoom in on the information as well as we can make sure you got the notes. Okay. And then here, you see those rectus muscles move in the eye, either superiorly, inferiorly, medially, laterally. And then we have the oblique muscles that rotate the eye. Okay. Now, the layers of the eye. We're going to start with the outermost layer. This outer layer is the white of the eye. This is the sclera, also called the fibrous tunic. It is tough and protective. So if you saw that dissection, if you didn't, go back and look at it. It's on the content page with the area where the eye notes are, or the special senses notes are. It's a little bit tough when I'm cutting through it, and that's because it's protective. Okay. And then anteriorly, it's modified to be transparent, which is the cornea. So here is our sclera, and if we follow that anterior modification, it's transparent here, which is the cornea. Okay, let's zoom in a little bit more here because this picture is also showing the conjunctiva. I don't think they actually label it, but you can see that it's being peeled away here. That's the conjunctiva. So this is the sclera here. Anteriorly, it's modified to be transparent, which is the cornea. Now, while I'm on this picture, I'm going to talk about the middle layer here, which is the choroid layer, which is also called the nutritive tunic. The innermost layer here is the retina, also called the sensory tunic. We'll talk about the modifications of those layers. This ball of gelatinous material, if you watch the dissection, you'll see me plop that out. That is called the vitreous humor. Okay. And then there's fluid anterior to that, and that's called aqueous humor. We'll continue and we'll talk about all these layers in a minute. Okay, so the choroid layer is, let's zoom in a little bit. The choroid layer is called the nutritive tunic. That is the middle layer. It has lots of blood vessels and is pigmented to prevent the scattering of light rays in the eye. Okay, now, on that dissection, when we got to that choroid layer, you saw that it was like a blue-green iridescent color. We don't have that. Animals have that. So if you have a pet and you've seen their eyes glowing at night, then that's what that is. That allows them to trap even small amounts of light so they can see better in the dark. Which I was walking in the dark last night in the hallway, and my cat was up under my feet, and of course he would scream if I stepped on him, but he could see, I couldn't see. He doesn't realize that. But that structure is called the tapetum lucidum, right? The tapetum lucidum is that iridescent layer inside that allows them to trap even minuscule amounts of light so they can see in the dark. We don't have it. So if you're in the house and a person's ass starts glowing, either you need to get out or they need to get out because you got a problem going on because people don't have that. Okay. Now we have some modifications to this choroid layer. One is the ciliary body. The ciliary body is a muscular structure that helps to regulate the movement of the lens. The lens is responsible for bending light or refracting light. So when light comes in, it needs to be able to flex or contract or relax. The lens itself doesn't do it, but the muscle does. To bend fibers of the lens for refracting or bending the light to go to the focal point in the back of the eye. So the ciliary body is muscular and it has these tiny fibers that extend from it called suspensory ligaments. Your textbook might say suspensory zonule. I like ligaments better because they look like ligaments. Okay, and I'll show you in a second. Then you also have the iris. The iris is the colored part of the eye that is muscular. Two different sets of muscles there that regulate the pupil, and the pupil is the opening through which light passes. And so if there's not a lot of light, like when I was walking through the hallway with the cat, my pupils no doubt were dilated to try to let in as much light as possible. And if it's a whole lot of light, they constrict. And speaking of cats, their pupil is not shaped like ours, right? So if you have a cat and you have this cat sitting in bright light, their pupil looks like a slit, right? Just a line up and down. But if you put them in a darker area, it dilates and it gets round. Okay, and that is because of the amount of light available. So the muscles in the iris, we got circular that constrict and narrow the pupil, and then we got radial that dilate and open the pupil. I don't know if that is in your notes, so if it is not, make sure you write that down. Sympathetic stimulation is going to cause pupil dilation. Parasympathetic stimulation is going to cause pupil constriction. So I want you to write that down too, and then we'll look at the pictures. So here, the blue here represents the iris, and the picture on the side is showing you the circular muscles and then the radial muscles and how they function to either cause constriction of the pupil or dilation of the pupil. Okay, let me see. I need to go back because I want to look at that choroid layer some more. So let's zoom back in on this picture. And I'm going to have to do it for the retina as well. Okay. So the choroid layer is this middle layer. It's kind of a beigey color here. Right. And then when we get here, it's modified. That's the ciliary body. Right. And so they label it here, ciliary body. They label the choroid here. You can see all these blood vessels going through it. Okay. Then you see the suspensory ligaments extending from the ciliary body to the lens. Okay. Also anterior to that, you see the iris. So this is, it looks like it's blue here. And then the opening in the middle is the pupil. Okay. Okay, now let's go up to our retina. So the retina is also called the sensory tunic. It is the most inner layer, and it is very delicate. So if you watch that dissection, you'll see it's super thin and it's kind of a consistency of wet tissue paper. So it's easy to damage. So people who have damaged retinas, they have laser kind of surgery to repair. But honestly, you can't really put wet tissue paper back together again. It's very, very delicate. And so we have photoreceptors, and I want you to take note of that term on the retina. And photoreceptors detect light. So we got two categories of photoreceptors. We got rods and we have cones. Okay. And rods, and they're named based on the shape. So rods are more cylindrical and cones are more conical. So that's kind of how they're shaped, and that's why they're called that. And so rods are for night vision, peripheral vision, black and white vision, and cones are for color. And so within rods, you have something called rhodopsin that helps for distinguishing or picking up the light rays that are associated with black and white vision, night vision, peripheral vision. And then the cones, you have photoreceptors that are different color cones: red, green, blue. Red are L type, green are M type, blue are S type. This probably isn't in your notes. You want to write it down. And they detect different wavelengths of light. Okay, and so we'll look at that. I need to go down in my magnification a little bit. And so we looked at yesterday. There's an area on the back of the retina where the optic nerve comes in. That's called the blind spot, or the optic disc. Well, it's called the optic disc, and we refer to it as a blind spot because at that place there are no rods and cones. And then next to it is an area called the macula lutea. And the macula lutea is the focal point. And in the center of that is the fovea centralis. Now, rods and cones are scattered all over the retina, but that fovea centralis is where we have a higher concentration of cones. So let's make a couple notes here. This macula lutea is a focal point. In the center of that is the fovea centralis, where we have a high concentration of cones. Okay, that color doesn't work well, so let me change the color here. We already said the rods are for black and white vision, night vision, peripheral vision. Cones are for color vision, and they're concentrated in that fovea centralis. Let's look at the picture. So you can see the rods and the cones here. The rods are obviously more rod shaped or cylindrical. The cones are more conical. So they are named based on that. Now within the rods, we have this rhodopsin, which is a membrane spanning protein here that is specifically the receptor point here. Now, you don't need to remember anything other than this name. You don't need to try to. There's a lot of information surrounding this molecule, and we aren't going to go heavily into that. Okay. Let's go back to my eyeball and look at the retina. So this lighter kind of orangey color kind of represents the retina. This inside, this is the layer here. Right here, and this is not a good picture of it, is the optic disc where the optic nerve is coming in. This particular picture doesn't show the fovea centralis. Hopefully, I have another one that will. If not, I think I have another set of slides pulled up that I will show you that on. Okay, so cones, we said you have SML cones. They're sensitive to different wavelengths of light. And short, medium, long wavelengths of light. So that's where we got those letters from. And so you can write down this information. I'm not as concerned about um, the nanometers of light. I just want you to appreciate that they detect different wavelengths of light, and then we're going to look at a picture that indicates the colored area of that the wavelengths of light that they detect. Okay, so shorter, you see that falls into the blue range, medium falls into the green range, long falls more into the red range. Okay. Um, Let me pause you for a second. I want to look and see, do I see something better that shows my fovea centralis and macula lutea. Okay, so my other set of slides did have this. So before I left talking about the retina, I wanted to show you this. So here where we come in, so all of this inner layer is the retina. This is the optic disc. The retina actually springs from the optic disc. So if you see the dissection, you'll see that once I move this whole retina around, there's a point where it's attached. That's the optic disc, where the optic nerve comes in. But just next to that is this macula lutea. It's kind of like a bullseye. And then the center of the bullseye is the fovea centralis. And that's right next to the optic disc. That is our focal point. Okay. They show a little bit on this picture. We can zoom back in and look at that picture. And they show a slight depression on this one. And you see that right next to the optic disc. So here's my fovea centralis in the middle of my macula lutea. Here's my optic nerve. Here's my optic disc, which is the blind spot. No rods and cones there. Okay, let's go back to my other set. Okay, so other structures, we got the lens that I said before is responsible for bending light or refracting light. And so you have the ciliary body attaching the suspensory ligaments on one end and the suspensory ligaments attached to the lens on the other end. Let's zoom in on this picture. So here's my ciliary body, right? And it goes around. So this is a different view. So you can see how it goes around like this. This is my lens, and these are my suspensory ligaments. So when this contracts and relaxes, it either stretches this lens out, right? Or it allows the lens to be more um, relaxed and thicker. And so um, it tells you here that if the lens is stretched out and thinned, that is for distance. And here, if the ciliary body relaxes, then the lens is going to be thicker and for close up vision. Okay. Now, there are some spaces in the eye. So you have an anterior segment and a posterior segment. The anterior segment is where we have aqueous humor. There's liquid there. And the posterior segment is where we have the vitreous humor. That's that jelly-like mass. Okay. The anterior segment has an anterior chamber and a posterior chamber. Anterior chamber is before the iris, and posterior chamber is after the iris. They are all filled with aqueous humor. That's the anterior segment. Posterior segment is the back of the eye where we have the vitreous humor. Okay. I'll zoom in on that. So how do we see things? Okay. So light passes through the eye, right? So we have light rays that come in, and these, let me make that straighter. The light rays come in. The cornea and the lens help to bend the light to the focal point. So we want the eye to hit the focal point. The light to hit the focal point. We then perceive that image. So what happens is the light comes in, the rays would be scattered. They would just go straight, right? If we didn't have the lens and the cornea, and so we wouldn't get a focus. Those rays would just, so the lens and the cornea help to direct the light, bend the light to the focal point. And so once that light passes there, then the image is formed on the retina. It is inverted, is carried to the optic nerve, and then we perceive it right side up. Okay. I'll give a couple definitions. We talked about the rhodopsin. Within the rhodopsin, we have opsin and retinol. So that information is down here, photopigments. And then we have a definition of accommodation. So our ability to change focus depending on where the location of the object is. I'll give you a minute to write that. Okay, so now our intention is to send all of the light rays to the focal point in the back of the eye. And under normal circumstances, we do that. But there are some conditions where we miss that focal point, right? Either we are not going far enough, our focus is before the focal point, which is myopia, nearsightedness, or our focal point is after, and I don't think it's on this one. Let's see. a minute. Let me zoom in. Okay. Hyperopia is where our focus is after. So you can see here, we're beyond the length of the eye. Okay. So either the lens is weak or our eyeball is short. In the case of myopia, our focal point is before we focus before we get to the focal point. And then the lens is too strong or our eyeball is too long in that case. And I'll zoom in so you can take a look at those. Astigmatism is where there's a curve on the cornea here. And we'll look at that. Presbyopia is age-related changes in vision. And that is often where people need reading glasses. The focus is beyond the focal point. Let's zoom in on all of these pictures. Let me delete that stuff off. Okay. So, oh, come on. This one is associated with changes associated with age, where you need reading glasses. The focal point is beyond where it would be. Here, these are showing you with and without the corrective lenses. So this is myopia, nearsightedness. The focus doesn't go all the way to the focal point. And then with corrective lenses, we're right at the focal point. With hyperopia, we go beyond the focal point, or what would be the focal point, right? So we don't see well. And then with corrective lenses, we're at the focal point. Astigmatism, you see there's an abnormal curvature of the cornea, and then the corrective lens will balance that out so that our image would be reflected properly on the focal point. I think those terms are already in your notes, but I'm going to zoom in a little bit so in case you need to write those down. Okay, then we move to the ear. The ear is for hearing and the ear is for balance. And we've already talked about the structures, so I won't go heavily into that again, but we have our outer ear that has our auricle, our auditory canal. Then we have our middle ear, and our middle ear has our tympanic membranes, our ossicles, and our auditory tube. And then our inner ear has the cochlea, the vestibule, the semicircular canals, and of course, the nerves. Okay. So the outer ear, the skin, the cartilage, that directs sound into the ear. In that external auditory canal, on the you have or external acoustic meatus, you have wax, right? That's called cerumen. And you have ceruminous glands that secrete that. That is protective, is to prevent stuff from getting down into the ear. You're not supposed to stick a Q-tip in the ear because you can jam that wax further into that ear. I have a brief story about that. Somebody, a really close family friend, was having a little trouble with her ear over during COVID. And she never could, she went to a regular doctor, but she couldn't get into the ear, nose, and throat doctor because of COVID. They weren't taking new patients. They were only seeing, their current patients with, serious issues. And so it wasn't that big of a deal. She would, I guess, feel a little pressure or something in her ear. And she didn't, it wasn't a big deal. It was just periodic. And so over that time span, she just let it go. And then it got to be where it was, irritating her a little more. And She called again. She still couldn't get in. So, they had given her appointments, and for whatever reason, they had to cancel the appointment. Blah blah blah. So she did have an appointment at least at one time. So she was at work doing some kind of training. She was the one training people, and she started talking like really off the wall, and they thought she was having a stroke. So they told her, "You need to go to the doctor." Blah blah blah. So she called her husband. Her husband came and took her to the ER. I think. But did she go to the doctor? I can't remember if she went to the doctor that time or not. But anyway, she may have, and they didn't do much, or what? Maybe she didn't. I can't remember that part. But anyway, she came home. She laid down. She slept all day and night. And so the next day, her husband was like, "No, you're going to the doctor." And um, so she was like, "Okay." She ended up driving herself to the doctor. However, it turns out, and the girl there was oh, I think that's what happened. Her husband brought her home. She just laid down, and she went the next day herself to the ER, and the nurse there um was helping her. She had a buildup of wax in her ear to get the wax out of it. So she kept flushing it or whatever she was doing, and she pulled out quite a bit of wax in her ear. She said, "Oh yes, that feels better." She came home and she wasn't better, and she was better for a time period. And she said, as soon as she got home, she didn't feel good again. And this is where she laid down. And her husband the next day, no, that night, it was nighttime. I don't know if it was that night or the next night. Anyway, as you can see, I don't remember the details of the story. She told me every single detail. I can't remember all the details of the story. But anyway, she got to the hospital. It was at night. And this second time. And they looked in her ear, and they immediately called in surgeons to do surgery in the middle of the night. So this had to be a severe thing for them to call in surgeons in the middle of the night. Typically they schedule surgery. So they did some surgery. They said she had a severe ear infection. At least that's the story we got. When they were like, "Oh, let's pray for her. She has this going on. She's in the hospital." And then they had to go back and do a second surgery, a brain surgery, because that infection had spread so far. And that's why she was talking off the wall and all this kind of stuff, because that buildup of wax had pushed down so far into her ear, and there was associated infection. So you have to be, my whole point of all of that confusing story was that you have to be careful about not um, jamming wax further down into your ear with Q-tips. There's oil you can put in your ear. You put a few drops, it'll cause the wax to start moving closer to the surface. You're not supposed to put anything beyond this point right here. So if you can take a warm washcloth and pull wax away like that, that's what you need to do. If you put some drops in here, it'll cause the wax to come closer to the surface, and then you can wipe it away. Some people have more wax buildup or production than others, and you can consult your doctor about how to best handle that. Now, I say that I use Q-tips. You're not supposed to use Q-tips, but I also don't produce a lot of wax. But you're not supposed to stick anything down into your ear. And actually, the package of Q-tips would tell you don't stick this down in your ear. It's supposed to only just clean around here. Okay. Anyway, that long, drawn out, confusing story was to say she had so much wax buildup in her ear, it was hindering her hearing, hindering the pressure in her head, and also had caused a severe infection where she had to have brain surgery. And it still is not fully recovered. And this was months ago now. She's primarily recovered, but she still has some memory issues and um, issues with drawing on what would be the right word. Like, she was explaining something to me and she couldn't remember the word vertigo. She remembered it begin with a V, but she couldn't remember the word. So those kinds of things. So you want to just be really, really mindful of that. Anyway, So the middle ear is where it's called the tympanic cavity. That's where we have the ossicles. The tympanic membrane leads to that space, and then we have the auditory tube that equalizes pressure. So when you go up in a plane, for example, and you feel you got pressure in your ears, and you chew gum or you yawn or you open your mouth real wide, you're rushing air through that tube to regulate the pressure. The inner ear. So now we have three areas in the inner ear: the cochlea, the vestibule, and the semicircular canal. So we're going to take them one at a time. So the cochlea is the part where we have the organ of Corti for hearing. This is snail shaped, and so they cut through it, and so then that's what it looks like. So then if we take one section of that out, then that's what this looks like. And then we take a middle section of that out, and that's what that looks like. So we looked at that on the model yesterday. So when we look at this part close up, we have the scala vestibuli, the scala tympani, and the cochlear duct or the scala media. These two are filled with perilymph, which is a fluid, and this one is filled with endolymph, which is a fluid. So I want you to write those down. Okay. Talked about them yesterday. The scala vestibuli and scala tympani have perilymph, and I have that term somewhere on here. I just saw it here. And in the cochlear duct or the scala media, we have endolymph, and I don't know if I have that on here, so I'll just type it out. Here. Okay. Um. Okay. So when we talk about how we perceive sound, let's zoom in a bit on the picture. These are fluid filled spaces. You have the stapes. Go back. You have the stapes sitting here on the oval window. You see that? Stapes is sitting on the oval window. Right. And so what happens is when a sound wave hits the tympanic membrane vibrates, the ossicles vibrate, and then it causes the fluid in these spaces to vibrate. Okay. And when the fluid vibrates, and it causes the membrane to vibrate, and actually this basilar membrane vibrates. And when the basilar membrane vibrates, have you ever been to a party where it felt like the floor was vibrating, like the floor was going woo, 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 with the sound of the music and the people? That happens. You've heard of parties on the news where the floor crashed in. Okay. But the same kind of thing here. So this kind of vibrates like that. And these hair cells stick up into this gelatinous mass called the tectoral membrane. And these hair cells, this one kind of holds the membrane in place. And then these cells detect the changes because of the vibration and send that into the cochlear part of the vestibular cochlear nerve for us to perceive sound. So those vibrations translate into this fluid filled space, cause the membrane to move, and these hair cells will detect that movement as they are projected into a gelatinous mass. Those are called mechanoreceptors. Let's write that down. mechanoreceptors, and I'm sure I probably have it on a subsequent page. Okay, so this is a close-up of what we just talked about. Okay, so I think I said something wrong the last time. I'll let you write that down in a second. Well, here these are the ones that are holding um, things in place, and then these are the ones that are perceiving the sound. Okay, so this is my basilar membrane that's going to vibrate. These hair cells are projected up into this tectoral membrane, holding that, and then when this vibrates, these inner hair cells will detect that vibration and send the information in to the vestibular cochlear nerve. Oh, didn't mean to do that. Move this down. Okay. And this might be different than what is said in the current notes. I think I changed it, but I'm not sure. So it's not the tectoral membrane that moves, it's the basilar membrane that moves. Make sure you get this down. Make a note. This might be different than your notes. And so, a couple definitions here. Pitch is determined by frequency. The more frequent the waves, the higher the pitch, right? So, if we talk about that, then we're looking at something that looks like this, right? So, the waves are frequent, and my waves are not nice and neat, but loudness, the size of the waves. So, the bigger the waves, the louder the sound, the smaller the waves, the softer the sound. Timbre has to do with the clarity. And then we can have deafness, right? The inability to hear. And there are a couple of different types of deafness. Conductive deafness. The sound waves are not conducted into the ear effectively. So, actually, something like that buildup of earwax can actually cause something like that. I know that sounds odd, but it can. And then sensorineural is where the sound waves are passed into the ear, but not converted into sound waves. Okay. So this little picture kind of shows you some causes or things associated with different kinds of deafness. So, um, ruptured eardrum, impacted earwax, conductive. Something is not allowing the sound to get through, right? And then sensorineural drug effects, something that cause damage like loud sounds in your ear, aging, right? Infections, head trauma, genetic disorders can result in either of those possibly. Okay. Now, our ear is also for balance or equilibrium, and we have two structures in the ear that allow for that. Now, these are also mechanoreceptors. So we have the vestibule, that's this middle area, and in the vestibule we have, let me change my color here. We have the utricle and the saccule. Okay. And they have a structure that looks like this, where you have um, kind of a membrane that these cells are sitting on. Again, they have these little hair kind of projections from them, and they sit in a gelatinous mass. This gelatinous mass also has crystals on top called otoliths. So when she moves her head, right, up and down, the crystals move and they pull the mass with it. And when they pull the mass, the gelatinous mass, then those hair cells move. And so you detect these kinds of up and down and back and forth movements. Okay. So that's called static equilibrium. And I'll zoom in in case you need to write that down. I'm sure that part is in your notes. And then we have the semicircular canals. And the semicircular canals are for dynamic equilibrium. So they detect angular acceleration or rotation of the head. And we have the semicircular canals. Let's zoom in on this picture. And three different planes, right? So here's one, here's one, here's one, right? And so you have what's referred to as the ampulla and the cupula for the semicircular canal. So at the base of each semicircular canal is this enlarged area called the ampulla. And inside of that is a gelatinous mass called the cupula. So when we look at that up close, you can see that again, we have hair cells, which are the mechanoreceptors, projected into this gelatinous mass, cupula. And in this space and inside of the tube, we have fluid called endolymph. So when you are moving around, like on a roller coaster, for example, this fluid moves. Right? Let's look at this picture over here. The fluid moves, the cupula bends, and you have a sense that your head is moving. Now, have you ever gotten on a roller coaster and you got off and you feel like you're still moving? Why? Because this fluid is still moving through your ear. So you have to sit still for a minute and it'll stop moving and you'll go back to feeling normal again. So this detects angular acceleration. Now, for taste, we have papillae on the tongue. Some of them have taste buds, some of them don't. Anytime you see that term papillae, it means finger-like projection. So we have filiform, which are the tiny ones that cover the entire tongue. They do not contain taste buds. Then we have the fungiform that are scattered over the surface of the tongue. I guess we'll say that they are medium sized. They contain taste buds. Then we have the circumvallate, the big ones in the back. They contain taste buds. And then we have the foliate, which are the wrinkles on the side, or the folds on the sides of the back of the tongue. They contain taste buds. These are chemoreceptors. Chemoreceptors. Let me put that term here. I'm typing in white. I can't see my spelling, so I'm winging it. Chemoreceptors. And I did not spell that right. So that's because I couldn't see. Receptors. So move that E in front of the P. Okay, and so these receptors are the taste buds within the papillae, and they're going to perceive chemical changes as these molecules dissolve in a fluid environment. So you have to have a fluid environment for taste and smell. So this is why if you're sick and your nose is dried out, your mouth is dried out, you have a hard time tasting anything. Gustation is the term for taste. Olfaction is the term for smell. Okay, so we perceive tastes of sweet, sour, salty, bitter, and umami, which is a metallic or a meaty taste. Um, and it used to be believed that you could only perceive these tastes on certain areas of the tongue. Well, I'm going to show you what those areas are, but this is not a hard rule, right? So there's a whole lot of overlap. You can perceive different tastes in different areas, but these are the areas that we originally believed to be the case. So sweet on the tip and the top of the tongue, obviously stimulated by sugary molecules like glucose. Sour on the sides, perceived by or stimulated by acids. Bitter on the back, those things that are maybe more alkaline in nature, various poisons, that is protective. So if stuff is bitter, you common sense would say, don't eat that, right? We still kind of eat it. Other animals have sense enough to avoid it. Let's see. I have a note here about nitrogen containing compounds that can be poisons that are tasted, perceived by those papillae in the back of the tongue, or the taste buds in the back of the tongue. Salty on the very tip, stimulated by sodium or like molecules. Umami on the back of the tongue by meat, right? Amino acids, meaty or proteins. Glutamate is one of the amino acids in particular. I will zoom in on that in case you need to get a closer up view. Okay. So here are those papillae. Let's zoom in on the picture. Right. Circumvallate, spongiform, filiform. Now, if we look closely at those, we can see taste buds. So taste buds are tiny and embedded in the papillae. And so you have, again, some slight hair-like cells, and you have to have these chemicals dissolved and perceived by these hair cells there. Okay. Let us see what else we got. Okay. So where do we, umami is more towards the back, but we got salty on the tip, sweet on the tip and the top, sour on the sides, bitter at the back. Okay, so they believe that us perceiving the taste of fats, this is not a hard and fast rule at this point, is stimulated by fatty acids. The idea is that maybe people who are more able to detect smaller amounts of fat are less likely to eat a lot of fat and less likely to be overweight. I don't know. So I must not have any of these fatty acid receptors because that's what I like is fatty stuff. And so here I am fat. Gut tastes. So there are cells in the gut that detect the chemical makeup of the stuff in the gut and stimulate, excuse me, certain aspects of the digestive process. Right? And I'm sure that's not in your notes. You can write that down. Last thing is smell. We got the olfactory bulb that sits on the cribriform plate, sends fibers through, and we create an olfactory mucosa. Again, you got to have a moist environment for those odorant molecules to dissolve on that mucosa. And smell is olfaction, taste was gustation. Let's zoom in on this picture. We've looked at that before. So here is our olfactory bulb sitting on the cribriform plate. The fibers go through, and we end up creating this olfactory mucosa, and our odorant molecules have to dissolve in that mucus. Okay. Now, I talk about something else called the vomer nasal organ, VNO. These detect pheromones, and pheromones are substances that we cannot detect. Like I can smell perfume, right? I can smell soap. I can smell, the cat box, right? You can smell different things. So you can detect that and you can respond accordingly. But there are things, chemicals that are released that we can't smell that our body actually responds to. A lot of these pheromones have to do with closeness to people, like uh, mother to baby, um, sexual partners, that kind of stuff. And these are undetectable. Okay. We have a small area that is our vomer nasal organ that allow us to pick up on pheromones, and we have a special area in the brain that detects these things called the limbic system that triggers emotions. And so our vasonasal, vomer nasal, I'll get it right in a second, is small comparatively. And I have this picture of this cat here because if you've ever seen a cat make this face, right? You can see little cats, house cats do it, or big cats, even lions do it if you watch those kind of TV programs. And they make that kind of crazy face. Their eyes look crazy and they're holding their mouth open. And what's happening is they have a large area that is the vomer nasal organ, and they can detect all kinds of pheromones. So theirs is much larger. And that's what they're doing there. They're perceiving things that are sent, signals that are sent to that vomer nasal organ. Okay. So that is all for those notes. Any questions, anything that I kind of, just like this vomer nasal organ, kind of didn't pronounce correctly initially, you have questions about or whatever, please email me. We can get together and talk about anything. So you have all the notes, you have all of the anatomy, you have everything you need to prepare for this exam. The exam is due during the holiday. If you are busy during the holiday, you need to do the exam prior to the holiday because you will not have an opportunity to do it afterwards. So make sure you do it prior to if you're not going to be available during the holiday. Okay. Email me with any questions that you have.